in the previous class, we were discussing on the microbial metabolism. So the definition and the various things that are associated with that. So as you are aware, uh, it is the chemical reactions that happens in a biological system. They are called as biochemical reactions. And you are aware that in a living cell, thousands of such reactions happen, chemical reactions. Different substrates combine to form an end product. So, which is mediated by a catalyst. It's similar to the one which you have learned in chemistry, where two substrates combine to form an end product, which has to be mediated by a catalyst. So, there in chemistry, you use catalyst. The same thing in a biological system, the chemical reaction that happens, you use the word biocatalyst. The biocatalysts are usually, in most of the time, they are the proteins. So they are called as enzymes. So the biocatalysts, they are the enzymes, and the chemical reactions that happen in a in a biological system are referred to as biochemical reactions. And of course, in a living cell, there are different kinds of things that happen. Two kinds of major reactions that happen in a biological system. One is you know two objectives, different things that go on. First thing, a living cell, for its sustenance, it needs energy. It needs building blocks for it to perpetuate, to reproduce, to produce new cells. And so there are two things. One is the energy metabolism, energy yielding reactions, and the other one by using simpler building blocks to produce different macromolecules which are part of a living cell, like proteins, lipids, carbohydrates, nucleic acids, and so on. And of course, in this microbial metabolism, you will learn both the things, the one which are associated with the energy metabolism. It is called as catabolism. It's actually, so the like an oxidative reaction. A reduced compound is oxidized in the process. It is realized in the form of the production of ATP molecules. On the contrary, some of these things which are produced in this catabolic oxidative reaction, degradation of the organic molecules, are reduced in inorganic molecules, as in the case of the chemoautotrophs or photoautotrophs, chemoautotrophs. And now some of these intermediaries may be reused for the like synthesis of the building blocks of these cells, so which is called as anabolism. So you have two kinds of reactions, the anabolism and catabolism. Anabolism, of course, it is uh, also called as biosynthesis of macromolecules, which is energy intensive. It requires energy for that. On the contrary, catabolism, which actually produces chemical energy currency, ATP molecules, which is used for the synthetic processes. So this is with this concept, the thing. And another thing, I have told you, I have reminded you, the sum total of biochemical reactions that happen in a living system. So it is called as metabolism. They are of two kinds: catabolism and anabolism. Catabolism, results in the release of energy, and anabolism, of course, it results in the biosynthesis of molecules which utilizes energy. Okay, and these reactions, they are actually, in this case, in any of these, you have two things, of course. They are oxidoreductive reactions. Most of these reactions, oxidoreductive reactions, and the one, which are the source of this electrons, they have to be in highly reduced state. The molecules which are in reduced state, they act as donors. The molecules which are in oxidized state, they act as acceptors. So electron donors and the electron acceptors. In between, you have the catalysts, biocatalysts, the enzymes. Okay, so all these biochemical reactions that happen in metabolic activities, whether it is catabolism or anabolism, they are oxidoreductive reactions. And of course, these oxidoreductive molecules, you call such molecules, we can think, like be in two states, either reduced state or oxidized state. So mainly because of this property, the electron which is generated in a particular thing, on the donor's end, it it goes through a series of molecules and ultimately to be accepted by an oxidized molecule, which is called as electron accept. It's also called as electron sink. So this is the thing, based on all these things, you classify the like metabolism, what kind of donors, what kind of the electron acceptors. Based on that, you classify broadly the things into the fermentation, and the other one is respiration. And today I have, like the slides which are of course, in addition to that, in the previous class I have explained different molecules which act as vectors, which act as, electron carriers. For example, many of them, they are, like nucleotide derivatives. So they are cytochrome molecules or non-heme iron proteins, or flavin molecules, and so on. And so they are the some of the examples, which you come across in different things, like pathways associated with this metabolism. So they are the carriers of electrons. They'll be able to they carry the electrons to the final electron transport system, so which actually happens across a membrane, bilayered membrane. In prokaryotes, it is the cell membrane which will take care of that particular thing, across the membrane. Whereas in eukaryotes, for that you have a specialized system you call the, mitochondria. Mitochondria in a cristae, across the double layered walls of these mitochondria, that particular thing, happens. So all that, and you know, we shall go through some of these slides, which are part of these things, So the basic concepts we have already defined: the metabolism process of, of course, sum total of all biochemical reactions that happen in a living system, and which are of two kinds. As I have already told you, catabolism, the process by which a living organism obtains its energy, and in the process, of course, the main objective of this process, catabolism, is to produce energy. At the same time, what happens? It has dual function. So that is the beauty of a living system. Here it also, some of the intermediates which are produced during catabolic, in the catabolic reactions, they act as raw materials, for the biosynthetic pathways. The next reaction is anabolism, the process by which the energy and raw materials are used to build macromolecules and cellular structures. It is also called as biosynthesis. Some of the things, for your example, biosynthesis of proteins, biosynthesis of lipids, biosynthesis of carbohydrates, and biosynthesis of nucleic acids. Some of the familiar things, that are so the be given as examples. Of course, as I have told you, all these reactions, the basic concept, it's oxidoreductive reaction. So in all these reactions, a reduction and oxidation occurs And the chemicals which are involved here are oxidoreductive chemicals, which can exist both in oxidized state or reduced state. So that, because of that, they can act as electron carriers or vectors. So an atom becomes more reduced when it undergoes a chemical reaction in which either it gains electrons, or secondly by bonding to a less electronegative atom, and thirdly, it can also occur when atom becomes bonded to a hydrogen molecule. All this you have learnt in chemistry. And the secondly, an atom becomes more oxidized when it either loses electrons, or when it is bonded with oxygen, or when it is bonded with a more electro positive atom. Actually, it should be a electropositive atom, oxidized. So of course, the some of the things which I have already explained. So the redox things, the in the metabolic pathways, that are things, the you come across various things, which can act as oxidoreductive molecules. Then the enzymatic pathways for the metabolism. The metabolic reactions take place in a stepwise fashion in which the atoms of the raw materials are rearranged, often one at a time, until the formation of the final product takes place. So that's the thing. You have seen how a glucose molecule, while passing through, for example, in the regular cycle, glycolytic pathway, and in aerobic organisms through TCA cycle, and ultimately passing through the electron transport system and to be accepted by the oxygen, the terminal electron acceptor. Each step requires its own enzyme, that you should know. So it is the multiple enzymes, multiple steps. That's why you call each step, it is similar to a chemical reactions. So which happens in a living cell, you call it as a biochemical reaction. So totally, to define metabolism, it is the sum total of all biochemical reactions so that happen. So each step requires its own enzyme, specific enzymes. The sequence of enzymatically catalyzed steps from a starting raw material to final end product is called as enzymatic pathway or metabolic pathway. So that's the, for example, that is series of reactions that you come across. Of course, the other things are cofactors and redox reactions that have things, Um, so of course, I have told you the cofactors, which act as electron, shuttle to electrons in the metabolic pathways. They are the one, which you have in your e-notes. You have in the previous class I told you. They include some of the things, nucleotide derivatives like NAD or FAD. These are relatively small organic molecules in which uh, part of the structure can be either reduced um, or oxidized. For example, NAD. NAD to, it is the oxidized state. NADH, NAD plus H, it becomes reduced. So NAD plus H or NAD, it is the oxidized state or reduced state. So thereby they are able to donate a pair of electrons. NADH, by donating a pair of electrons, it comes back to its oxidized state, NAD, or similarly FAD, or in other molecules, various, cytochromes or non-heme proteins and various things, which you have learned in the page number 29 in the e-notes, metabolic pathways. There are two main redox things, I have told you, the NADs and the FADs. Of course, other than that, there are other things also. Of course, just now I have explained how, which, they can be in both states, oxidized state or reduced state. NAD or FAD, it is the oxidized state. By adding electrons, protons, H plus, and a pair of electrons, they get reduced. So NADH or FADH. So this is the things, Yeah, so thereby, they can, they are they can be in oxidoreductive state. They can be either in oxidized state or reductive state. That's the things, thereby they'll be able to, by uh, losing electrons, they get oxidized, or by accepting electrons, they get reduced. That's the things, so that you're aware. Then another thing, of course, in all these things, um, we talk about the energy, how the energy in a biological system is realized. It is realized not like a thing, paper currency that we use. It is realized in the form of a chemical energy currency. That's chemical energy currency. How it is realized? It is, of course, most universal thing. Of course, it is ATP, adenosine triphosphate, which is also referred to as currency of energy. You know, which is required. Which is used in many of the cellular reactions. Of course, similar to ATP, you can also have GTP. Of course, that you say in TCA cycle or many other reactions also you come across. But the most things, common molecule that is thing, that is seen in most of the metabolic, it is the ATP. So this ATP stands for adenosine triphosphate. It's a nucleotide with three phosphate groups linked in a small chain. So three phosphate groups, of course ATP, its previous state it is ADP, and it is the basic state is AMP, adenosine monophosphate with single phosphate bond. By adding one more phosphate bond, it becomes ADP, adenosine diphosphate. By adding three phosphates, which is the ultimate thing, the state of the molecule, it is adenosine triphosphate. So which has three phosphate bonds. So the AMP becomes ADP. ADP, of course, it will never reach, most of the time AMP, it, it happens, conversions between ADP to ATP, ATP back to ADP. Another thing, here you should know, ATP, when there ATP is there, where is the need for ATP? ADP. ADP is must, because like similar to NAD or FAD. So which actually the, by accepting electrons, they become NADH or FADH, and again by losing it, they become their oxidized state. So that, thereby they make themselves available for accepting the electrons further. Similarly, ATP, when it is hydrolyzed, by losing one phosphate bond, it becomes ADP. This ADP, once again, it's by accepting a phosphate bond, it becomes ATP. So this particular thing, for addition of phosphate bond, it is in biochemistry, it is referred to as phosphorylation. Phosphorylation. Uh, the next thing is, of course, the ATP. In case of ATP hydrolysis is used as a energy source in many biological reactions that require energy. For example, active transport in the sodium potassium pump, varieties of things, that you are aware. And in catabolism, energy is released from the oxidation of carbon is captured and used to synthesize ATP from ADP and phosphate. So how it happens, it is given here in the form of a chemical reaction. So a molecule of glucose along with 6 molecules of oxygen and 6 molecules of carbon dioxide and 6 molecules of water and and ADP and phosphate and energy, ultimately it gives rise to ATP plus water. So along with releasing ATP, at the end of the reaction, the water molecule is released. So till this particular thing, we were discussing how it happens, in the metabolic pathways, like in the catabolic pathways, in the biological system. Based on the things, we have defined the metabolic pathways into two broader kinds: the fermentation, where both organic molecule, both electron donors and electron acceptors are organic in nature. That's first point to be called as fermentation. Both electron donors and electron acceptors have to be in organic, state. Organic molecules act as both electron donors and electron acceptors. Acceptors, then it is called as fermentation. Another point that is a very important point associated with fermentation is the electron acceptor. It is it comes within the things system, that is endergonic. It is internal, it is generator internal. It doesn't come from outside source. Secondly, in case if it is the electron donors, they can be either um, you know organic or inorganic molecules. But the electron acceptors, in case if they are inorganic, then you use the word respiration. So in case of fermentation, both electron donors and electron acceptors are organic. In case of respiration, the electron donors, they are either organic or inorganic, but the electron acceptors have to be inorganic. So further, based on the what kind of what is that electron acceptor, further in this slideshow, you will be able to see that particular thing. So what is that? The electron acceptor. In case if it is oxygen, you use the word respiration, aerobic respiration. In case if it is other than oxygen, like oxygen containing ions, molecules like nitrate, sulfate, and other things, then you call it as anaerobic respiration. Anaerobic respiration. So in this case, of course, you are aware, we have discussed in some time in theory class, the glycolytic pathway in the catabolic reactions. You come across two important things, pathways. The first one is glycolytic pathway. The second one is TCA cycle or citric acid cycle. The first one, glycolytic pathway. Here what happens, the glucose is partially oxidized, and at the end of that, the pyruvic acid is the end product. Pyruvic acid is the end product. Then it is called as glycolytic pathway. Of course, in this there are alternate pathways. So there are things, there are different kinds of glycolytic pathway. So by definition, to call it as glycolysis, it is the conversion of glucose to pyruvic acid. Pyruvic acid is the end product of glycolytic pathway. Then you should be able to realize, in which organisms they have this glycolytic pathway. So it is found in all organisms, irrespective of whether they are, aerobes or anaerobes. In all organisms, this glycolytic pathway is found. Found. Further, of course, I will explain, how, it is different in aerobic organisms, the further reaction. So the first one, in the catabolic reaction, it is the glycolytic pathway. The feature of this glycolytic pathway, partial oxidation of glucose to form pyruvic acid. And in the process, glucose is a 6-carbon compound, pyruvic acid is a 3-carbon compound. So from a molecule of glucose, you will get 2 molecules of pyruvic acid, and in the process, small amount of energy is realized in the form of ATP. In the process, a small amount of NAD is reduced to NADH. Of course, that NADH that is formed in aerobic organism, it is realized in the form of ATP. It gives rise to 3 ATP molecules. But in anaerobic organisms, like which have fermentative metabolism or anaerobic respiration, fermentative organisms mainly, so that NADH, that is formed, it is of course reconverted to NAD to make the things go on the metabolic pathways. So the overview, what happens here in this particular thing, glucose, ultimately it results, it is shown in this picture, 2 molecules of pyruvate or pyruvic acid. So, and in the process, 2 ATPs are produced, of course, per each molecule of things, so there are 4 ATPs and 2 NADH are produced, but 2 ATPs are utilized in the during the process. Thus, the net yield of this reaction at the end of the glycolytic pathway, 2 ATPs and 2 NADH. So this the thing, three reactions alter the range of the 6 carbon glucose molecule into 6 carbon fructose 1,6 phosphate. Of course, further all that, in the glycolytic pathway, in the things, you'll be able to see the pictorial things of that. Okay. So the by definition, in your by definition, glycolytic pathway, it is the conversion of glucose to a partial oxidation to convert it to pyruvic acid. So from a molecule of glucose, you will get 2 molecules of pyruvic acid with the the end products are two net ATP production and 2 NADH production. And this glycolytic pathways, there are 4 major alternate pathways, glycolytic pathways found in the bacteria. The first one, which is very familiar and found in all living organisms, Embden-Meyerhof-Parnas pathway, EMP pathway. This is called as, it is the classic glycolytic pathway. It is Embden-Meyerhof-Parnas, EMP pathway. It is also called as classic glycolysis because it is found in all living organisms, all living organisms other than microorganisms. In the other animals and plants, it is found. So and among the microorganisms, it is found in almost all microorganisms. And other than this EMP, you also have other 3 alternate pathways. They are hexose monophosphate pathway. It is also referred to as pentose phosphate pathway, PPP pathway, pentose phosphate pathway, PPP, or hexose monophosphate pathway. It is also found in most of the organisms, responsible for the synthesis of pentose sugars used in nucleotide biosynthesis. This is very important. This HMP pathway or pentose phosphate pathway, the main purpose of this, because it is found in all microorganisms, it is the production of pi-carbon sugar. Pi-carbon sugar, ribose and ribulose, which are actually required in the synthesis of DNA and RNA. Ribose or ribulose. So that particular thing, so the pi-carbon sugars that are used. Then the second thing is Entner-Dudroff pathway, or it is also called as ED pathway, which is found in Pseudomonas, the genus Pseudomonas, which is a gram-negative bacterium. So it is in this bacterium and related genera, this ED pathway is found. Then the next pathway is phosphoketolase pathway. Phosphoketolase pathway, it is found in Bifidobacterium bifidus or Leuconostoc, mesenteroids, and other species of Leuconostoc, the 2 genera of lactic acid bacteria, which are also referred to as heterofermentative lactic acid bacteria, because they produce a mixture of organic acids, these bacteria. So the 2, of course, you can say the major picture from the thing, E nodes, and some of the salient features that happens. The first pathway, EMP pathway, Embden-Meyerhof-Parnas pathway, what happens? A molecule of glucose gets converted to 2 molecules of pyruvic acid. Initially, 2 ATPs are consumed. When towards the end, 4 ATPs are produced and 2 NADHs are produced, and ultimately net production is, so from a molecule of glucose, 2 molecules of pyruvic acid and 2 molecules of ATP and 2 molecules of NADH are produced in this particular reaction. Then the hexose monophosphate pathway, microorganisms, main purpose of this hexose monophosphate, that is HMP, It is also abbreviated as HMP pathway or pentose phosphate pathway, PPP, PP pathway. So here what happens, the glucose, of course because they are all alternate glycolytic pathway, you are aware by definition their aim is to convert glucose to pyruvic acid, with the production of NADH or ATP. But here slightly different from the classic EMP pathway in that when glucose gets converted into intermediaries are produced, you can refer to any like the pathway here at the end of the things, you'll be able to see that, or otherwise the on Google, you can search that, or microbiology books, you can search Google, it is available. And main thing here for you to remember, the HMP or pentose phosphate pathway, one of the key intermediaries here is phosphogluconate. And in this particular thing, remember, for you to remember, in the process, so many intermediates are produced, which are either 5 carbon or 4 carbon, 6 carbon or 12 carbon compounds. And from the 6 carbon or 3 carbon compounds, it can again go back to EMP pathway. It may follow the classic And the next pathway, it is Entner-Dudroff pathway, which is seen in the Pseudomonas and related organisms. Here again, at the end of the things, pyruvic acid and NADH are produced, but it takes a as like slight deviation, and here again the glucose is converted to phosphogluconate. In the previous things you have seen, the hexose phosphate pathway, phosphogluconate is also produced, but the next step, you will come across so many other things, this 4 carbon, 5 carbon, 6 carbon intermediate goes through in which pathway or pentose phosphate pathway. In this Entner-Dudroff pathway, the phosphogluconate that is produced, it gets converted to glyceraldehyde 3 phosphate and a molecule of pyruvic acid. This glyceraldehyde 3 phosphate, which is an intermediary of the EMP pathway, again it from there, from this stage, it follows the EMP pathway, and another molecule of pyruvic acid and NADH, pyruvic acid and NADH. It doesn't alter the chemical things. So the next pathway, phosphoketolase pathway, which is seen in the heteroactive bacteria like Gluconostat, Bifidobacterium, Bifidus, organisms. And in this, once again, of course, you will come across phosphogluconate as one of the intermediaries. Then it leads to the production of a 5 carbon intermediate plus carbon dioxide, and this 5 carbon intermediate, so it breaks down to yield a 3 molecule glyceraldehyde 3 phosphate and a 2 molecule acetyl coenzyme A. This acetyl coenzyme A, it can right away, it can enter the TCA cycle in the microaerophiles or aerobic organisms. It has that ability by combining, fusing with a molecule of oxaloacetic acid. Acetyl coenzyme A yields a molecule of citric acid. So like there, this glyceraldehyde 3 phosphate that is realized, again it follows the classic EMP pathway from that stage onwards to yield a molecule of pyruvic acid, NADH and ATP. These are all some of the things that slight alternate things that you come across in this glycolysis. Glycolysis, by definition, it is the partial oxidation of glucose to produce two molecules of pyruvic acid, that is three carbon compound pyruvic acid, with by realizing a small amount of energy in the form of two ATPs and a reduced coupling agent, that is NADH. Here we should slightly alter to have a glimpse of the fermentation, because in the organisms which possess fermentation, which possess which follow fermentative metabolism, in these organisms, how it happens. Some of the examples, like the organisms which produce alcohol, which produce lactic acid, which produce different other organic acids and so on. So what happens in these organisms? So by definition, fermentation, we are aware, where the electron donors and electron acceptors both are organic, and the electron acceptor, this is a very important point for you to remember, it is endergonic. It is generated internally, you must remember. It doesn't come from outside, and it has to be organic, and it is generated within the biological system. So the features of the fermentative pathways, and here the fermentative pathways, the beginning of this fermentative pathways of all the organisms which follow fermentation, the glycolytic pathway, it is common in all of them, it is found. And the from the stage of pyruvic acid, the end product pyruvic acid, so from there on, depending on their metabolic makeup, so whether one organism may be an alcohol producer, another organism may be a lactic acid producer, another organism may produce butyric acid, and so on. Depending on that, from pyruvic acid they follow different pathways. They side reactions. What happens? Some features: the pyruvic acid is reduced to form reduced organic acids or alcohols. Of course, that just now I have told you. The final electron acceptor is a reduced derivative of pyruvic acid. That you must remember. The electron acceptor, so the molecule which acts as electron acceptor in this process, fermentation, it is one of the reduced derivative of pyruvic acid. In the further you will be able to see that. Then another thing is the NADH, that is oxidized. Here what happens, the NADH, it is oxidized, which is formed during glycolytic pathway, because they are the end products. End product of glycolytic pathway, it is pyruvic acid plus ATP plus glucose, NADH. This NADH, which is produced in glycolysis, it is oxidized to form NAD, which is essential for continued operation of the glycolytic pathway. Otherwise, what happens, if no NAD is available, glycolytic pathway can't continue. So there should be continued supply of NAD. That's why the NADH is reconverted, reoxidized to NAD. Then it doesn't require oxygen. Then no additional ATP is generated in this fermentative pathway. So other than the two ATPs, production of two ATPs, then the gas, carbon dioxide, hydrogen, or may be released in the processes fermentative pathway. In majority of the fermentative reactions, CO2 is the common end product, and hydrogen is occasionally produced. Then fermentation pathways are useful as tools in biochemical identification. That's very important. Especially, this is you will learn maybe in this week the next practical topic, that is biochemical reactions of microorganisms. There we will how the fermentation products they act as indicators for a particular organism. The fermentative pathways are useful as tools of biochemical identification. For example, carbohydrate metabolism. When a carbohydrate it is fermented. You'll get organic acids. You will get many intermediates, all that. That particular will they are used as tools for the identification, biochemical identification. Then next point, they are also used in industry. That's a very important thing. Fermentation from the point of energy metabolism, because the net production in fermentation, it is only 2 ATP molecules, because NADH that is produced, it will never be realized in the form of ATP, because it doesn't go up to the electron transport chain. So but it is reused in the process. So that is nullified. It's only net production of 2 ATPs. So in the fermentation, that's only the energy that is the produced. No more energy can be things. But the other very beautiful aspects of fermentation, the organisms which follow fermentation, their end products have industrial applications. For example, alcohol, industrial solvents, and alcoholic beverages like beer, winemaking, distilled alcoholic beverages, then ethanol production, and so on, and various organic acids. They are utilized in the biosynthesis of organic acids like citric acid, gluconic acid, and so on, malic acid, and so on. So they are very, very important industrially. The next thing, examples, of course, where the thing, some of the examples for fermentation pathways. The lactic fermentation found in many bacteria like lactobacillus, then streptococcus, pediococcus, leuconostac, bifidobacterium. All these organisms, they follow the, of course, they follow ferment. But the one, pediococcus, the leuconostac, bifidobacterium, they are heterofermentative lactic acid bacteria. They produce only traces of lactic acid. But this lactobacillus, the genus lactobacillus, there are many species here. And the streptococcus cremoris, or others, these two lactic acid bacteria, the bacterial genera, they are the classic examples for the homofermentative lactic fermentation. That is the lactic acid producers. So they are the main things used in the biosynthesis of lactic acid. Then mixed acid fermentation, just now we were discussing. Of course, the other two, bifidobacterium and leuconostac, that's in heterolactic fermentation. Similarly, in Escherichia coli, so other than these things, other organic acids, they are also produced, the Escherichia coli. So, for example, this particular mixed acid fermentation, it is utilized in the identification process. That is MRVP test. You use a test called in biochemical reactions, methyl red Voges-Proska reaction. That particular test, here B it is given here, basis of methyl red test. So you will classify whether they are acid producers or neutral product producers. The next thing is 2,3 butane fermentation. This is the one, many organisms like like you have Enterobacter. That is, earlier it was referred to as Aerobacter aerogenes. Now it is referred to as Enterobacter aerogenes. This particular organism, Enterobacter aerogenes, it is known to produce this particular key compound 2,3-butanediol, which is a neutral product, which is utilized in a test, MRVP test, where the second part Voges-Proska reaction. In that particular reaction, you will use that. We will study in the next in the practical class, that is biochemical reactions of microorganisms. Next, these are all the two classic examples, the fermentative organisms, how they are very, important industrially. For example, the first group of organisms, they convert the end product of glycolysis, pyruvic acid or pyruvate, to lactic acid, where the NADH that is produced, along with pyruvic acid, that is reoxidized to NAD, NAD, the lactic acid. This is very important. Then ethanol, of course, this is, of course, as source of this lactic acid, lactate or lactic acid, is one classic example. Another classic example, the organisms which process this particular reaction. For example, they are lactic acid bacteria, either homolactic fermenters or heterolactic fermenters. They are very important in many things. For example, in the preparation of curd, in the preparation of cheese, in the preparation of yogurt, in the fermentation of vegetables, like for example pickling, sauerkraut making, and so on. So the in all these reactions, these lactic acid producing organisms are utilized. The second group of such organisms, they are ethanol fermenting organisms, which actually, from the end product of glycolysis, that is the pyruvic acid, they convert it into ethanol. Here what happens? The acetaldehyde, which is produced as an intermediary. So pyruvate is reduced to acetaldehyde. This particular thing, this acetaldehyde, it acts as electron acceptor to uh, to form ethanol, ethyl alcohol. And the organisms which, the alcohol producers, which have the ability to convert glucose to the alcohol, ethyl alcohol, that is through pyruvic acid as an intermediary. They are very important. They are utilized, of course, in the production of bioethanol, in the production of like the spirits, the which are utilized in the preparation of beverages, distilled alcoholic beverages, or in the preparation of beer fermentation, or wine fermentation, or pure alcohol production. They are utilized. And of course many more things. This alcohol, they have the importance. These organisms are mainly represented, like by yeast. Of course, many bacteria are also there. Whereas the first group, lactic acid fermentation, is mainly dominated by the bacteria. And the mixed fermentation, of course, where the from a pyruvic acid, which is the end product of glycolytic pathway. So along with the another end product, that is NADH, it gets converted to lactic acid and the NAD on one side. And from this pyruvic acid may pass through a series of things, organic acids like acetic acid, formic acid, along with the production of hydrogen and carbon dioxide. And ultimately sometime this carbon dioxide may also be converted to carbonic acid. And these are, of course, this particular mixed reaction is found in heterolactic fermenters like bifidobacterium and leuconostac. And it's also found in the Escherichia coli, E. coli. And you're aware, the in hot summers, the curd, it doesn't have the normal the flavor, aroma of the curd, lactic acid. Sometimes it gives the thing, flavor of tastes differently. The reason is that, many, some of the things, many times, these contaminant organisms may carry over the other reactions with the production of acetic acid or formic acid, or even alcohol, to give the curd different taste. That you must have you have realized in hot summers many times. Your curd or buttermilk tastes differently. So then the butanediol fermentation, which is seen in Enterobacter aerogenes. Here what happens, the pyruvic acid, it is converted to initially acetolactic acid and carbon dioxide. This lactic acid, it is converted to AMC, that is AMC, acetomethylcarbinol, AMC. It is abbreviated as capital AMC, and it is also called as acetoin. This acetoin or acetomethylcarbinol, it is converted into 2,3-butanediol, which is a key compound utilized in Vogt-Prusker reaction, which we will discuss when we take up that things, practical portion. Uh, then the importance to give you some of the important things, importance of this fermentative organisms. Of course, the ethanol production by Saccharomyces cerevisiae, or propionic acid production by Propionibacterium. Of course, one of the species, Shermani, Propionibacterium shermani, which is used in the preparation of Swiss cheese, Swiss cheese, with characteristic eye buds and the propionic acid flavor, bitter flavor, so which is actually seen in Swiss cheese, a class of cheeses. In cheeses you have like Roquefort cheese, Camembert cheese, Swiss cheese, then so many cottage cheese and so on. The processed cheeses, they are like the Roquefort cheese, blue cheese, or Camembert cheese, it is pink cheese, or this Swiss cheese, which is actually with eye buds characteristic. That is the propionic acid producing bacteria are responsible for that. Then the another thing is, these fermentative organisms, they are also utilized in the industrial production of acetone or butyraldehyde and butanol, and so many, where the obligate anaerobic organism Clostridium acetobutylicum is utilized. And at the end, you'll be able to see some of the things, how the end product of glycolysis, pyruvic acid, by the fermentative organisms is converted into many useful things, like for example the yogurt, or you know
Sir, sir,
ah
sir, sorry to disturb, sir. Time I.
ಟೈಮ್ ಆಯ್ತೇನೆ.
ಆಯ್ತು ಸರ್.
ಟೈಮ್ ನೀವು 12 ಕಾಲಿಂದ ಒಂದು ಕಾಲಲ್ಲೇನೆ.
ಅಲ್ಲ ಸರ್, 12 to 1 ಸರ್.
ಮತ್ತೆ ಹಾಗಾದ್ರೆ ನೀವು 11:45ಗೆ ಜಾಯ್ನ್ ಆಗಿಬಿಡ್ಬೇಕು ನೋಡ್ರಿ.
ಆಯ್ತು ಸರ್, ನೆಕ್ಸ್ಟ್ ಟೈಮ್.
ಯಾಕಂದ್ರೆ ಐ ಕಾಂಟ್ ಅಫೋರ್ಡ್ ಟು ಲೂಸ್ ಕ್ಲಾಸ್ ಅಲ್ವಾ.
ಸರ್.
See, I have to make up my ಈಗ you should remember, know that thing, like so the I should get my time. Okay.
Okay sir.
ಸೊ ಆಗ್ಲಿ ಕ್ಲೋಸ್ ಮಾಡೋಣ. ಇದೊಂದು ಸ್ಲೈಡ್ ಆದ ಕೂಡಲೇ ಇಲ್ಲಿ ನೋಡ್ರಿ, ಈಗ in these fermentative organisms, how they are economically important, like in the production of lactic acid, here in the production of dairy products like curd, yogurt, then cheese, and so on. And also they are also important in pickling industry, sauerkraut making industry. Next, ethanol production. They use alcohol producing organisms are important here, um, in wine production, beer production, alcohol production, ethanol production, and so on. Then butyric acid, Clostridium acetobutylicum, they are utilized in solvent production, butyric acid, butanol, acetone, isopropanol, and so on, various industrial solvents. Then propionic acid bacteria, it is utilized in the preparation of the thing, like Swiss cheese, a characteristic cheese. So it is. Then mixed acids, this acetic acid, lactic acid, succinic acid, and so on. It is once again it is utilized in the MRVP test. Then butanediol, once again in MRVP test. So with this, we will close at this. Tomorrow we will continue. What is the timing of the class, Mr. Ravishadesh?
Sir,
tomorrow
2:15 to 3:15, sir.
So you should think immediately before 2:15. So I will just like all of you should promptly join before 2:50. Okay.
Sir.
Okay. Thank you all. See you tomorrow.
Okay, thank you, sir.
Thank you, sir.